Hi, this is Roger Green, host of the Surfing the NASH Tsunami podcast. This weekend, we're offering 5 conversations from episode 60, our special coverage of Madrigal Pharmaceuticals' release of MAESTRO-NASH Phase 3 data, originally posted this past Wednesday. This conversation, the last one from the larger group, starts with me following up a couple of earlier comments. First, the fact that in other diseases, combination therapy can get approved before each element of the monotherapy has been approved individually. And second, that semaglutide's Phase 2 result, with its 72-week effects on steatosis and NAS activity score, but not fibrosis regression, aligns neatly with Scott Friedman's earlier question about the relationship between fat lowering and fibrosis, given that not all fat lowering may mean the same thing in the liver. Jörn, Schattenberg, and Mazen Noureddin follow up with other comments about NAS, fibrosis, and lipotoxicity, after which Jörn notes that there's already a second THR-beta in trial from Viking Therapeutics and asks what this trial might mean for Viking. Mazen goes on to cite Terns Pharmaceuticals as another company with other drugs, including a THR-beta in pipeline, and believes Terns will benefit as well. Scott Friedman asks what all this means for cirrhosis, and Rachel Zayas, in response to a question from me, discusses the development work her company is doing and how these results might affect her work and ability to raise resources. As we wind down, I ask the group what topic we've not covered yet. Kris Kowdley describes one more way that the use of companion diagnostics against dose response and non-response will aid payers. Louise Campbell discusses how we might consider this drug a major anti-cancer advance, given that liver is implicated not only in hepatocellular carcinoma, but in non-hepatic cancers. And each group member shares one closing takeaway. Here in Nashville, where we all live, the world changed for good on Monday. This is a quick take on some of what that change might look like. It will be interesting in years ahead to look back on this conversation to see what we got right and wrong. And you get a sneak peek right now. So sit back, listen, enjoy, learn. And when you're done, join the conversation in our LinkedIn discussion group.
Mazen Noureddin.
There's tremendous excitement today, and I've consulted for companies that they intentionally did not want to go to NASH because they said the investors are tired and fatigued and frustrated. So yes, sponsoring is important, and to me, I would like them to pour some of that money in patient advocacy, awareness, in addition to just investments and stuff like that.
So a couple of questions or naive comments, or both. Question/naive comment number one. I made the statement on the podcast several months ago that you could not get a combination drug approved until the individual elements have been approved, and we got feedback that isn't accurate in antivirals and in some situations in oncology. So it seems to me that it's certainly not reliable in those cases. It's not clear to me whether that'll turn out to be the case in NASH or not. I mention that because I think it's Terns whose strategy is about going straight to combination. And I know THR-beta is one of the things they're looking at to go straight to combination in, and I doubt, so we may actually see people pursuing that path as a way to market. Thought number one. Thought number two, going back to something Scott said earlier, in combination with what Mazen just said. So SEMA has a 72-week trial that shows effect on NASH activity score, but not on regression of fibrosis. I think that is the single thing that walks me right next to where Scott was when he talked about how compelling it was that this drug proved it could do both, or that it could regress fibrosis without having direct anti-fibrotic activity, because it says that there's something different going on here than what goes on with at least that GLP-1.
You're right. I think also Scott was commenting about the type of the fat that needs to be melted as steatohepatitis. I thought it was a brilliant comment that depends on which type of fat, and Jörn added to that lipotoxicity versus not. So yeah, I do agree with that, and I do agree that it's encouraging data for semaglutide. We should recall the data from the NASH CRN that showed clearly that if you reverse NASH resolution, or you achieve NASH resolution or 2-point NASH improvement, you will eventually get fibrosis improvement. It's just a matter of time. So yeah, all great comments.
Jörn Schattenberg.
It's a good comment, and it feeds back to the theme of personalized therapy, and obviously the presence of lipotoxicity as a driver of the disease to be addressable by the mechanism of action of the drug that's required. Still, as you said, it's rightful that the that trial has started recruiting in the US, and Europe is going to be starting up soon. So that is important. One more thing I had on my mind we haven't discussed yet. I found interesting that there's actually a second compound in class. So we're discussing out-of-class combinations, but there's a second thyroid hormone receptor beta agonist, and it's gotten a little more quiet around it. I think the company is called Viking, and what could that mean for other? Do we need something in the same class to be developed, and see how those data are replicated, being different drugs? And go ahead.
Good point, Jörn. I think Terns is actually at great advantage of the results today. They have multiple drugs in the pipeline. They have THR-beta, they have FXRs and others. I do think that, sure, I mean, we would like to see more drugs and see their efficacy and all this. We still don't know if other THR-betas will achieve what Resmetirom achieved today. I do encourage other research in that area, and that incredibly important receptor that proven to play a big role in NASH.
Scott Friedman.
Yeah, there's one other element that you embedded in your question, Roger, which is, or maybe you didn't, but I inferred it, is what about the unmet need in cirrhosis? Because to me, that's really the most urgent unmet need, because those are patients who are rapidly moving on to transplant lists or developing liver cancer. And yet they lose many of the features that seem so ripe for benefit from resmetirom, like reduction in fat, improvement in glucose homeostasis. Many of those patients are already losing weight. And so, correct me if I'm wrong, Mazen or Jörn or Kris, Madrigal has a cirrhosis trial ongoing. Personally, I wouldn't be surprised or disappointed if it wasn't nearly as effective in cirrhotic patients as in patients who have more full NASH with fat and inflammation, as much as I hope it is effective, because it may well be that there are different therapeutic targets that are critical to try to improve fibrosis once the liver has already gone to a cirrhotic stage.
I think you will find that it's easier to get investment now into other THR-betas, because at least you've now proven that one should be able to get over the finish line. If there's a second one being studied and the entrepreneurs or a company, Viking being a small company, can make a compelling case that theirs might work better on any dimension that matters, that's easier money to raise than what everybody's trying to do for modes of action that haven't been demonstrated yet, even if they can prove staggeringly good results.
Yeah, well, there's a long history of first in class is not best in class. It started with statins, or at least that's the most vivid example, but it also worked in hepatitis B and C antiviral therapy.
And that's only because you live in hepatology, Scott. I will tell you that the only time in medical history outside of oncology, maybe even in oncology, up until 2015, that the first drug in was the market leader on the day it went generic was Prozac. And that's across all cardiology, lots of things. Just someone always came up with something better. Rachel, anything that you're working on, looking on right now that has any bearing on the question of what might be under the hood here? And what subtleties might be embedded in the questions people have been asking? And you can say no, by the way. That's a wild card question.
Rachel Zayas.
I would say yes. And actually, we're at an interesting point internally in the company, because, of course, we need to have for context for everybody on this call and all the listeners, my company is developing 2 non-invasive diagnostic tools, one that can differentiate steatosis from NASH, and a second tool that can stage fibrosis from a targeted epigenetic sequencing assessment. So the first question is linking each of those to biological relevance. What's happening directly in the liver? How do we know that this is coming from the tissue of origin and associated with a biologically relevant assessment? And for context of use, we initially started developing these tools as diagnostic focuses, but then it's something that within our pipeline we're looking at if we can also monitor therapeutic efficacy and which drugs and how relevant are these to specific therapies. So that is something that we are grappling with internally and something that we're really interested in conducting. So we're talking to some places right now that I'll keep internal at this point, but this is definitely relevant. And as a geneticist by training and as who has a focus in genetics, a lot of this is really underexplored territory, and I think we're just starting to understand. So one thing is, we were just discussing, first in class is not always best in class. So what we're doing is we're trying to get our MVP to market and then further develop that to make sure it is the best. So a lot of synergies here with what we're doing as well.
We're almost an hour into this, which is usually the point in time at which we start trying to move towards the end. But before I even think about moving towards the end, all of you, what itch haven't we scratched that you folks came out of today's announcement with?
Kris Kowdley.
One of the most important downstream benefits will be if these results are consistent over time from a standpoint of non-invasive tests, since we have such a rich plethora of non-invasive tests. And this is a trial where the non-invasive tests done in parallel that includes both MR as well as transient elastography and liquid markers, if you will, show a relationship between dose and response and dose and effect, then I think that will also be a very important shot in the arm for those of us who believe that although liver biopsy is a valuable tool, is not ultimately how we're going to be helping this get into the hands of patients. And so the companion diagnostics used here from a treatment standpoint, from a point of view of payers, are going to be very critical. And I think we need to look at not only dose response effects, but also non-responder data, so that we can determine early on which patients are unlikely to benefit and then move on to other therapies. So I think the non-invasive testing showing in parallel similar results is very encouraging, and I think we need to really dive into that a lot more.
I'm glad that Kris mentioned this. I was, as you know, before I presented data from Resmetirom on something I really worked on for the last 2-3 years, and which is the MAS score, which consists of PDFF, MRE, and AST. And in a previous oral from the Resmetirom, we showed that MAS dropped with the Resmetirom compared to the placebo. So I'm anxious and curious to see if that MAS score will predict response to therapy. So I look forward to see that.
Roger, something that resonated with me also was in the follow-up questions by the investors, whether this wouldn't be now the right drug for all patients F1 through F4, opening this up. And the rightful answer was, of course, this trial was conducted in an F2, F3 population, and this is the label how it's going to read out. And I think we got to, with all enthusiasm, and I felt that enthusiasm in that question, why don't we give it to all, be reminded to really look at the data, be reminded on the patients that were enrolled, and I mentioned the metabolic risk factors, and that's important if we want to achieve those response rates as they have been reported. So while I could understood that person's question, why not give it to everybody with NASH, it's important to be remembered that the efficacy was shown within that inclusion-exclusion criteria, and we're not ready to just leave that yet.
I think, by the way, some of that was a confusion on the part of the questioner about the idea that 5% of that population was F1. And Becky Taub then actually explained why an F1C patient might have been included. So I think, first of all, you and I are in violent agreement on this, and my guess is so is everybody else on this call. But I think that small set of F1 patients might have teased an investment analyst in a direction they just shouldn't have gone. And that was my read on what that was.
Louise Campbell.
Can I just give you one more point? If we take this and it gets approval, and then we look at the long term, this could just be one of the biggest anti-cancer drugs to be developed. Because NAFLD and NASH and the cancer-driving mechanism of non-cirrhotic NASH driving the cancer and liver cancer outbreaks at the moment around the world, this has the potential to turn around to be the biggest cancer investment that we could make by treating these patients. So if this really is as good as we're making it out to be today, we haven't even touched on the potential of reversal of liver cancer for these patients in the future. And that is just momentous.
Much more to say there, but I'll climb up in the interest of time. But a great point.
Well, actually, I just wanted everybody's closing comments. So I'm going to treat that as Louise's. And then, Scott, if you would like that to be yours, go ahead.
Just dig deeper, find the science behind this, explain why this drug worked and in whom it worked.
Mazen, if you got to jump, go next.
Congratulations to Madrigal. Thanks for what they have done. Congratulations to the patients, most importantly. Thanks to all those that underwent liver biopsy. We promise you we'll take this away one day. and look forward to see the future with the combination therapy and additional efficacies.
Yeah, an important milestone in terms of now we have 2 trials that have shown positive results in phase 3 and a trial that showed both NASH resolution and fibrosis improvement with a challenging regulatory pathway with regard to liver biopsy. So I think it in some ways reinforces that drug development in NASH is possible, can be done properly. This is going to lift all boats. I think it's very encouraging for patients, given that we have this huge epidemic that's already upon us in terms of fatty liver disease and the downstream consequences.
Mike Bottel.
I just want to say that Stephen Harrison's picture of the graveyard, I'm so tired of seeing that negativity that this is finally an opportunity. Madrigal, congratulations, because your tombstone will not be added to that graveyard, and that's awesome.
So, the article that drove the graveyard might have been authored by a good friend and colleague of our podcast, Dr. Schattenberg, but we're going to save that thought for Professor Schattenberg. We're going to save that thought for another day. Okay, Rachel.
Well, I want to say thank you to the patients who participated in this clinical trial as well as various others. Thank you to Madrigal. And I just want to echo Louise's comment that this might be the biggest anti-cancer approach that we will have in for decades to come as a field. So I'm excited to see additional endpoints from a field perspective to further extrapolate the implications that this has. So thank you for the opportunity.
And just thinking of all the investigators that are also contributing, the patients. I mean, this means you keep your patient in a phase 3 trial, even if you're not the highest specialized NASH center, only running NASH trials in that trial for such a long time, which is essential for those positive results. Congratulations to all these patients that stuck to it. And we're going to be reminded it's an ongoing blinded study, and we're looking at the top line data here, and it's just a big excitement from my end to learn and grow with that data.
Louise, you have one more?
No, I agree with my colleagues, fellow surfers, new to the surfboards, old surfers, you name it. The future's looking bright, and I did a hepatitis C one. It's not necessarily the first wave that you're on. It may be the second, the third, or the fourth wave that you catch that makes it the best wave.
In this case, 52 weeks on the way to 54 months. But so I come out of politics in the first place, most of you know. Deep Throat said to Woodward and Bernstein, follow the money. And because patients were generous with their livers and investigators had the faith to pursue, we're now to the point where I think we can stimulate, as Kris points out correctly, this is the second trial that had efficacy, significant efficacy in phase 3. We're now to the point where we can stimulate enough investigators to create enough money to, as Scott said, follow the science. There are systems that are self-amplifying, and you need moments when the system amplifies. The rules change today. Just a very simple level. The rules change. Stephen described this day as a watershed. I thought Yasmin Rahimi's question on the podcast this morning, where she said when she read the press release, it brought tears to her eyes, because God knows she invested in a lot of companies that didn't get there, or had her people invest in a lot of companies that didn't get there, or might never get there. All the rules change for all of us today. And so I think it's just a banner day for all.
And now, back to Roger.
We hope you've enjoyed this recording. If you have any questions or comments about the content of this conversation or the entire episode, please send an email to questions@surfingnash.com. We will be back next week with the rest of our year-end reviews. 2 episodes that include conversations with Mazen Noureddin, Ken Kuzi, Jeff Lazarus, Stephen Harrison, Naeem Al-Khouri, and Ian Rowe. You'll want to hear all of it. Until then, wishing all of you a wonderful holiday season and best wishes for a fantastic 2023. Stay safe, surf on. We'll see you next year on the podcast. Bye-bye now.